Today we will discuss about this intracellular signaling when the immune cells interact with the antigen, and we will cover in this video about B cells and T cells intracellular signaling, and we will also understand how these T cells differentiate into these Th2 cells or T helper cells type 2. In our previous video we have understand how to distinguish between Th1 and Th2 cells. In this video we will understand how these T cells differentiate themselves into these Th2 cells, which has this tumor progression properties. So we will start with these B cell receptors. So when the B cell, we will start with these B cells. So in these B cells, when the antigen interacts with these B cell receptors, it will trigger this signaling pathways, that is PKC zeta, and that will leads to the activation of this ERK12, and this ERK12 will cause phosphorylation of this IK beta pathway, and that helps in this complex formation, that is P50 REIA and IK beta phosphorylated, and this complex formulation will cause NF kappa beta to translocate into the nucleus and upregulates the antibodies that are generated against the antigens that is been recognized by the B cell receptors. And simultaneously this ERK12 pathway, what it will does is it will also translocate this CFOS into the nucleus, and that will leads to the upregulations of the cytokines such as interleukin C and BCL-XL that will cause apoptosis. So it will depends on the kind of antigen that are being recognized by the B cell receptors, and based on that it will trigger either apoptosis or interleukin 6, and these signaling pathways will be interrupted by this PAR4 that will inhibit this PKC zeta activation. So this is about the B cell receptors intracellular signaling whenever there is antigen will interacted or recognized by this B cell interact with this B cell receptors. Now we will understand how the T cells differentiation occurs. So in this T cell differentiations, when the antigen interact with this T cell receptors, it will trigger these two pathways. It is PKC lambda by zeta and this PKC zeta, and both of them will cause activation of this IKK pathway, and that will lead to this phosphorylation of this IKB, and a similar complex formation occurs, that is IKB P50, IKB phosphorylated P50 and REIA, and that will cause NF kappa beta to translate into the nucleus and upregulates the genes that are involved in this interleukin 4 and interleukin 2. Simultaneously it will also helps in this Th2 differentiation. So this is not only the reason why these cells are T cells differentiate into Th2. There are other pathways also that also helps in this T cells differentiation into this Th2 cells, and that is this PKC lambda by zeta will also cause the activation of this JNK pathway, and that will cause translocation of this NFATC1 into the nucleus, and that will also helps in the differentiation of these T cells into the Th2 cells. And apart from this PK lambda by zeta and PK zeta, another pathway will also get triggered, that is MALT1 and TRFA6 complex formation will occur, and that will interact with this P62, which will also helps in the enhancement or activation of this IKK pathway. And apart from this antigen interaction from the T cell receptors, there is another receptors, that is interleukin 4 receptors. So when the T cell receptors receives either the antigen or the interleukin 4, then also it will do the similar functions. That is, it will activate this PKC zeta pathway or PKC lambda by zeta pathway. However, this PKC zeta pathways are functions or Intracellular signaling is similar to these T cell receptors, but there is a slight variation when the PKC lambda by zeta pathways are activated by interleukin 4 receptors. It will leads to the activation of this JOC1 pathways that helps in the complex that will trigger when the complex formations occurs between ZUC1 pathway and phosphorylated STAT6, and this complex will then move into the this complex will then help the nucleus to helps the genes helps the transcription of the genes that leads to the differentiation of the T cells into this Th2 phenotype. So, and PKC zeta pathways has two functions. It will activate IKK pathway, and also it will activate this complex formation, that is JAK1 and STAT6. So when the interleukin 4 receptors uh, receives the interact with the interleukin 4, then it will trigger both the PKC zeta pathway and PKC lambda by zeta pathway that will have the similar effect in the downstream signaling pathways, that is this complex formation, and that will lead to the Th2 differentiation of the T cells. So I hope you got the basic idea about this intracellular signalings when the immune cells interact with the antigen or the interleukin 4 cytokines. Now this is not the only signalings that get activated. These are the some of the signaling pathways that are being identified. If you need to understand more about this area or this topic in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.